All right, hi everyone. Let's get started. So welcome to the Reverse Metabolomics workshop. I'm Vincent. I'm a postdoc in the Peter Dorenstein lab, and I'm going to be presenting with Yasin, also from the Dorenstein lab, and Santosh from University of Turku, and then Prajeet from the Dorenstein lab. We want to make it as more interactive as possible, so if you have any question, please just raise your hand or just ask directly the question. You can also disturb your neighbor, so you're just welcome to disturb whenever you want if you have any question about any of the sections. So let's get started with reverse metabolomics. So reverse metabolomics is, for example, if you have any molecule that you're interested in, right? So it could be a class of molecule that you're interested in. It could be a molecule, a known molecule. It could be a non-known molecule. It could be a molecule that is higher or is significant or it seems to be involved in a particular disease, or a molecule that is specifically accumulating in a different organ. And then you start with the MSMS spectrum of this molecule. And then you might be interested to know what is the organ distribution of this particular molecule, or where is it found, or which organ is it found in. You might also want to evaluate the distribution of this molecule in mice versus human, for example. Or you might want to know if the mice model or a pig model would be more adapted to study your particular molecule of interest. You might also want to know if this molecule that you just recently discovered or that you're interested in is also associated with specific disease. So is it found in cirrhosis? Is it found in Alzheimer's disease? So these are the type of questions that you might be interested in and which reverse metabolomics can help you. Reverse metabolomics is way more versatile than this, but this is just a starting point for you to try to learn and then dig into the reverse metabolomics. We also have different domain-specific masks that allows you to tackle different questions. So if you found a feature into a mouse model in, let's say, the liver, you might want to know if this specific molecule is actually produced by specific microbes. And for that, we have micromast. So it's 60,000 LCMS run. It's monoculture of microbes. And then you might find this molecule in specific monoculture. We also have plant mast, so plant-derived molecule. We also have food mast for food-derived molecule. And we are currently building microbiomast, and I'm going to talk a little bit about it in a few minutes. So today is actually to provide you with enough information to get you started. So we don't want you to be expert in reverse metabolomics and be super fluent, but we want to give you the basic to understand it and be able to do it by yourself. So today, the workshop is actually meant to query a small number of molecules, let's say 3 to 5 molecules, but we already gave everything available on the GitHub to query 10 to hundreds to thousands of different molecules and to associate all these molecules to different body parts, for example. So we just published recently a paper in Nature Protocol. It's a guide to reverse metabolomics, so it's a step-by-step. So if you don't understand fully everything that is happening today, you can just refer to that protocol. It's a guide step-by-step, and this allows you to perform reverse metabolomics. And reverse metabolomics is 4 different parts. So the first part, you start with an MSMS spectrum. You perform MAST, so it's going to search in the public domain where is this MSMS is found in. You're going to associate and integrate redo metadata to it. Yasin will tackle that part. And then the part 4 is the validation step. We're not going to talk about the validation steps today. And the reason is this step is dependent on your research question. So we just not going to address that for today, but you're welcome to talk with us about it if you're interested in. And what is reverse metabolomics? So it's a data science strategy that will take the MSMS as the first approach to search public domain and the public domain, and then to associate metadata. So we already published 3 papers that are leveraging that same tool. So the first one was by Emily Gentry, where she used reverse metabolomics to discover new bile acid and link that to patient with inflammatory bowel disease. We have a second one where we discover a new class of bile acid that are linked with polyamines, and they were associated with diets. And then we just published a recent one in Cell here, where we use another class of compounds called anisolipids and link that to different body part specific diseases, and we found them, some of them are actually produced by specific microbes in your gut. So these are the 3 papers that for now we have published leveraging that specific tool. And for today's presentation, we're going to be focusing on 1 bile acid, so phenylalanine cholic acid, and 2 anisolipids, so histidine conjugated to C4. and then phenylalanine conjugated to C4 as well. So these are the 3 molecules that you're going to go through with you today and associate body part distribution and if they are found in specific disease and if some of them are actually produced by specific microbes. Again, reverse metabolomics compared to traditional is traditional, you start with a defined question. So you have a predefined question you want to address. You design your experiment, you collect the sample, you run the sample, you generate the MSMS spectra. And in reverse metabolomics is a little bit the opposite way, right? So you start with an MSMS spectrum and you want to know in which data set or where this molecule is found in the public domain. So you use MAST, our search tool, that's going to search across all 3 main metabolomics repository, and then it will retrieve identical or similar MSMS spectrum, and then you integrate metadata, and then you can ask different questions, for example, different intervention, organ distribution, life stages, disease, and there are much more information embedded into the metadata that you can just conduct yourself. There's 2 ways you can, there's multiple ways actually that you can retrieve an MSMS spectrum, right? So one approach that we used in the past was to use a synthesis-driven reverse metabolomics where we did hypothesize that some molecule might exist in the biology. We synthesize those, we acquire the MSMS spectrum, and then we start to search the public domain. The other way that we also tackle is MassQL. So here you can find specific pattern in the MSMS spectrum. So for example, for the bile acid, we know they have 2 dianistic fragment ions depending on the hydroxylation state. So if you're focusing on the 337 or the 319, then we know those belongs to the trihydroxylated bile acid. You can use that tool to collect MSMS spectra that has this specific pattern in the MSMS. You can also use MSMS that you have on your computer that is part of the supplementary information of a paper. So there's multiple ways you can collect an MSMS spectrum and then launch reverse metabolomics with it. And then I'm gonna just pass it to Yasin.
Okay, hello everybody. So I'm going to talk a little bit about redo, because everything that Vincent was just talking about is basically about taking some MSMS spectrum, searching that against all the public MSMS spectra, all the public data that we have out there, which are billions of spectra, and then integrating that with the metadata that we have available for these different sample types. And that, of course, requires metadata. And what I'm going to talk about now is a little bit about how we even get to have this metadata, how we have all this raw data, and then also how you guys are able to contribute to this resource, because yeah, that's the only reason this works. Before I jump into that, I want to quickly check, because I know there are internet problems here at the venue right now. Do you have internet on your laptop? Because this will be at part hands-on. I know that some people have and some don't. Maybe a quick raise of hands, who was able to get internet access? Okay, so maybe all look around a little bit, because there will be some hands-on part, and then you can maybe move next to somebody who does have internet and then work together on some of the things where you can do something hands-on. So yes, with that, so to this slide a little bit. Does this laser point actually work? Okay, like this, perfect. Okay, so if you've been in the previous session, one thing you have heard is that there are a lot of metabolomics repositories out there. So here you have some of them listed. So GMBS, Metabolites, the National Metabolomics repository, or Metabolomics Workbench. And so, yeah, quite diverse resources out there. And what we have been doing in PanRedo is basically bringing all these different resources together and creating thereby one central resource from which we can utilize all this data that are out there and also integrate it with the metadata, so you don't have to necessarily go to each individual one if you want to find specific samples and work with them. And that allows you to do all these things that Vincent is going to talk about now throughout the next slides. So we have also published a paper on that I've put here on the left side. You also see that there's quite diverse sample types. Different repositories have different focus for what types of data that they're collecting. And yeah, but these are all things you basically don't have to think about if you just go to PanRedo, because we have aggregated everything together there. If you have your computer with internet connection, one thing you can do is click on this link up here. Did you all get access actually to the presentation earlier? Anybody does not have access to the presentation yet. There was a tiny URL in the beginning. Otherwise, I might just quickly go back and let you type that again. So there's this tiny URL in the bottom. Maybe type that into your browser. And then you should get access to all the material. I will give you a minute to do that. Oh, was it too fast? Sorry. Oh, that's the wrong direction. Wait on. Oh, okay, it goes in circles. Oh, that's interesting. Okay. Now. There we are. Okay, finally. Anybody still trying to get the tiny URL, or do you all have it? I see nodding. Okay, great. Okay, great. So afterwards, just go to this slide and click on the redo link, or just type it into your browser. Here on the upper, does this actually work too? Ah, yeah. Up here. Anyone still trying to get there and not there yet? No. Okay, great. So this dashboard that you're seeing here, that's the redo dashboard. So the way you can think about this is basically just all the metadata that we have been able to aggregate across all the public repositories that are out there. So on the side, you see some summary statistics on the number of files that are there, number of different sample types that we have available right now. And most importantly, you also see, not on my screen, but on yours, if you have opened it, some download button where you can just download the whole thing and work with it locally. With you, I just want you to play around. Actually, can't do it here because we don't have browser access here, I think. But oh, actually, let me try. Ah, no, it does work. Okay, perfect. So one thing you can do here, for example, is checking here the columns you have available. So I want you to go down here and select, for example, MS2 spectra count. Then go to the very left here. And then I want some files that have more than 2 MS-MS scans, or basically any MS-MS scans. And what I could do then is just select one of these files. Any, it doesn't matter which. And then you can just click here, for example, on this view or download raw data in browser button. And once you do that, you can now look at this raw file in your browser. It will take a little bit to download it, depending on the size of the file that you have selected right now. And once everything has loaded, you will be able to see this data file in your browser, and you can play with it without having to download it to your computer. And it's also interactive. I won't go now in detail through this dashboard, but yeah, you can basically look at different data that we have here directly in your browser. And you can do all kinds of cool stuff with this kind of data. Wait, how can I go back into presentation mode? Okay. Yeah, you can do all kinds of cool stuff with this data. Vincent is going to talk about quite some of them. So here's just like one illustrative example so you understand a little bit where this is going. So here you can see all the different organisms that we have in Redo currently, and you can now, with this MS-MS matching strategies that Vincent is going to talk about, start mapping different molecules to all this public data. So what I've done here, for example, is take these different molecules, map them against the different organisms we have available there, put it on this phylogenetic tree, and you can see that we have hits at very specific places here, and you can make cool findings like that by basically integrating, in this case, taxonomy with MS-MS matching or metabolomics data more specifically, and then make cool findings like that. Like here, for example, artemisinin, that's a malaria drug, and we find it in some specific plant, or like cocaine. Here we can find out that this is also in specific plants, but we can also find it up here in human. And we can make these kind of findings and observations and see where these different data show up. That's actually not what I'm going to talk about now. More specifically, I want to talk about some more boring things and leave the more interesting things to Vincent afterwards. And these more boring things is, how is this even possible? Like, how can we make these types of analysis, and how can also you make this kind of analysis? How did this become possible? Possible? And this is probably one of the more important buttons here, which is contribute your data, because all of this data that you have just seen now in this tree that was displayed, and all the data that you will look at later together with the others are public raw data. So people uploaded their raw data that they have measured, and now we can reuse them and look with a bird's eye view on all this metabolomics analysis that have been done in the past, which are,
We have published metabolomics data and placed it in Zenodo and raw files and extracted files.
At the moment, Zenodo is not one of the places that we directly support. We are working on that, though. So that will be one of the things that we hope to also index at some point. At the moment, what we index is just these repositories that you see here, which is Workbench, Metabolites, GMPS, and Norman. Those are the ones we have indexed up to now. Zenodo is, some things work already with it, but most things don't yet. But it's something we are hoping to also achieve,
As far as I've understood, once I've had it on Zenodo with the DOI, I can't put it in any of the other databases, right? Because it would be doubling it.
I wouldn't say you can't, because it's your data, but what you're allowed to do with your specific data files, I can't tell you. But generally, I would say the goal is to not replicate data too often, because it's like storage space. But I don't think it's a problem to duplicate it, usually, so at least in these cases.
Direct upload capability to your platform.
Great question. That will be the next few slides. Yes, so there are different repositories. I'm going to talk now mostly about GMPS Massive, because that is ours. There are also others. I will also refer to them later again, and it doesn't really matter where you upload your data. I will just show you how to do it in GMPS Massive now, and this will just be a very, very quick walkthrough. The main reason I put it here into the slides is because I want you to know how to do it. You have access to the slides, and I want you to have some basically step-by-step click through how you can do that. That's not really a hands-on thing now. I just want you to have this material to then refer to it if you're trying to upload your own raw data. I will just rather quickly go through it. So for GMPS, we host our raw data on Massive, which is mostly a proteomics repository, but it's also used for metabolomics. So that's where you upload your raw data. There's this nice submit your data button, but before you can actually click that, that's something that's not completely intuitive. You have to actually upload your raw data via an FTP client. So we typically recommend this WinSCP manager. You can just download that. You have it then locally. Then there are some steps you can follow, basically filling things out the same way that I did here, but with your user credentials. These are now mine that I have put here. And once you did that, you can just basically drag and drop your files into that. And then you will have access to this raw data or whatever data you have put there within Massive, which is then, let me use this again, which is then through this button here. So then you can submit your raw data through that, which basically just means you need to fill out this form here where you give some very basic information on what species is it, in this case Homo sapiens, which instrument did you use, no PTMs since we are not proteomics people here, and some other basic information. And then after clicking on any of these buttons here, you can basically just select your raw data, which you will now have access to in this file browser here in this small window that opens once you click one of these buttons. So here, for example, you can see some of my raw data. So I would now just, in this case, for example, click on the MZ.xml folder, because that's where I put all my MZ.xml files, and then click here on peak list files, and then I will do the same with the vendor raw data, because it's always good to provide both, and click on the raw spectrum files button, and then after I finish my selection, I can just submit it, and that will send my raw data online, which then everybody can have access to once you make it public. After this, it's actually not public yet. There will be another step, but that's pretty intuitive after that, to make sure they're actually public and also reusable by other people and your reviewers of papers, and so on. Metadata we collect at another site, also for the reason that many people first only want to upload the raw data very often and only later add the metadata. So we collect metadata at a different place, which you can find through the link I showed you already before, this redo GMPS link, and there's this nice contribute your metadata button, and that will then basically guide you through how you can submit your metadata. Also here, we'll just very quickly click through it. So it will bring you basically to a web page that looks like this, and then after you click on this button here, you will get to a nice Google Sheet with some instructions of how you can fill out this metadata sheets, what terms are allowed, what terms are not allowed, and descriptions for different things. Also here, some more instructions for you to play with later. There's also a validator implemented in this, which is then basically telling you after you have filled out your sheet, you can use this current sheet validator, and it will then tell you by coloring the different terms whether you did things right or not. So yeah, that's some quick check that I typically also do when I fill out the sheets, because it's very easy to make mistakes there. You will get then some summary report that just tells you what terms you did wrong. So here I put in some random missing values and X values and wrong term, and it will just tell you what things I filled out wrong. And you should then correct before you upload it. You can just download it as a TSV file and then submit it through the original portal. It was just a very quick walkthrough, as I said, and promised, I think, in the beginning, if I didn't forget, I will do the boring part, so no worries. Now you will talk again about molecules and MS-MS matching and how to make cool discoveries. But yeah, please keep in mind, the only reason these things work is because people upload their raw data and metadata to different places, which then allows you to do this kind of analysis, us to do this kind of analysis, you to look at your own data from a different angle, because you can very often make different findings when you use these kind of tools and put your data in context with other public data than when you look at your own data just with your in-house tools. You will just typically, as I think we all know, use like maybe 1% of your raw data, and the rest will just get kicked out because you can't really make sense of them in Metabolomics. And this will just allow you to get a different angle, and yeah, also the whole community to grow with that. That's basically the idea. And it doesn't really matter, maybe as a final point, which repository you use for loading up data. We are trying to always make use of everything. And yeah, with that, I will again move on to more exciting stuff about how to make findings, I think with Vincent again, right? Yeah.
Thanks, Yasin. Yeah, again, this is only possible because of the metadata and because the metadata that you upload, right? So again, just make it this is super important to share of that, and also for your type of research, and hopefully you see the benefits of it. And everything that it's in Redo is also embedded into those domain-specific maps. So if you have, again, those specific question of, is it produced by a microbe, is it coming from plant, is, let's say, from a different diet or food, this information is coming also from Plant Redo, right? So if you put your metadata there, then even yourself can explore your own data in a more deeper depth. As Yasin mentioned, they start to aggregate terms together, right? And then body part distribution, disease, and many, many more. But there's things that are not captured yet within PenRedo, and we're always trying to improve and capture more data and more information. So if you're thinking about it, there's no information on diet. So if you have a specific diet that contains beef, tryptophan, high-fat, low-fat diet, these terms are not yet captured within PenRedo. If you have a specific mice model that you're studying for, let's say, Alzheimer's disease, 3-XTG, 5-XFAD, those models are also not captured within Redo. So there are a lot of biological intervention or contexts that are not yet captured within Redo, and now we're creating a new ontology to improve the metadata capture more information, right? So for example, here we're building this new tool called Microbiome Mass, where we try to accelerate microbiome research. So what we're trying to do here is to bridge microbial metabolite to host biology. So if you know the microbe is modifying this type of drug, you find it into a study where you give this drug to a patient, can you rapidly identify which microbe can be doing that transformation, right? Which allows you eventually to mine, let's say, okay, this gene might do this modification, and then that might interfere with this specific treatment. So we're creating this resource now where we, let's say the first part here, where you have healthy versus disease, again, that are different terms that are not captured within Redo. You can have a probiotic intervention, you can have a surgery, you can have early life, aging. These are still not captured yet, but now we will be capturing them. We have different culture of microbes as a monoculture, but also as a community, right? So the microbes that are living inside you, they're not living together, right? They're interacting with each other. They're called communities. And what we did here is to build synthetic communities. So we know which member are part of the communities. We have all the information about those microbes at the gene level, right? So all these organisms have been fully sequenced, and we assemble them, and then we culture them with different type of molecules. So we have nasal, we have skin microbiome, we have gut, and then eventually we're developing one from urine. And we cultured those different synthetic communities with different building blocks. So we cultured them with different bile acid, with 140 different drugs, the mostly prescribed into North America. And we also are planning a big culturing experiment with 300 food biomarkers. And the goal here is to identify those microbial metabolites, so how the microbes are actually modifying those different molecules, and where are they reaching, on which organ they're reaching, how are they distributed, in which specific disease they are found in. And those specific, the type of analysis that you can now do is, let's say this microbe is modifying this drug, is it associated with non-responders versus responders, that you can try to improve different drug treatment. And we embedded all this new information within Microbiome Mass. It's not yet available, but it will be very soon. So this is the new, what you have here. It's the different origins, so if it's mouse, human, rats, different bacteria. We also have space sample, we also have pig, different animals, and we have also in vitro protein screening assay. You have different biological contexts, different interventions, so it could be a lot of different genetic mouse model. You have different diet intervention. You have a lot here from the mice. You have different type of infections. You can have viral infection, parasite infection, bacterial infection, different diet study in human that are already embedded here, and we have different surgery that have been performed where we have collected data on, and then so on. So there's a lot more information that's going to be embedded into Microbiome Mass. And just to give you one example, here we have N-acetylspermidine, a bile acid that was recently discovered by us in 2019 or 2022, I don't remember. So it was recently discovered, and then we didn't know if it was microbial derived. So if you use our tool that is called Microbe, you get that the mass was actually successful, but you get no matches. And then if you use tissue mass, that it's currently available also to you to use, where you try to contextualize molecule from different body part in human or in mouse, you can only have one hit here, that it's found in feces of different mice. So it's not very informative. I mean, this is the information that we have right now, right? But in the new interface that you're gonna all have access very soon, then what you have is this new tree here that we created, this new ontology where we curated. So if you go to the bottom right, you have the origin, then you have a specific microbes, and then we know which specific microbes is producing or conjugating N-acetylspermidine to this specific bile acid. And maybe these specific microbes are actually not in micromass, right? So it doesn't mean that because we don't have a hit here, that the microbe cannot make it. Here it's a control environment where we have different time points, so we know by confirmation in the experiment that we did that actually this microbe is able to produce this specific one. And we can start to contextualize this specific finding here, where you can have different hosts, and then it's found in mice and different intervention. We have diet intervention, we have sleep deprivation study, we have bacterial infection, It's also fine in rats. So now you can start to contextualize and then formulate way more hypotheses by using this new tool compared to the previous one. This is just one example, but there are many of them that now we know, okay, this microbe is modifying this drug and it's found into those patients that are prescribed, and then you can start to analyze if it's more associated with responders compared to non-responders. And I'm gonna pass it to Santosh, who's gonna just start briefly, and then we're gonna jump into the code and the script, the hands-on part very soon.
Thanks, Vincent. So now we will start hands-on for the reverse metabolomics part. So as we already learned from Vincent that the first thing that we need is an MS spectra. So that is the first part of reverse metabolomics. And in second part, what we do is like we source the we do the mask, that is we source in the public data. Then we match with the redo, and of course the fourth part is validation, which we are not going to cover here. So basically, like today we will be discussing part 1, 2, and 3, which is more on hypothesis driven approach. So for this to begin with, what we need is an USI, which is a universal spectrum identifier. So basically, like today we will be showing you using three of these conjugated compounds, basically like starting with the phenylalanine cholic acid, which is a conjugated bile acid, and then you have to repeat this with phenylalanine C4 and histidine C4, which is a NSI lipid. So what is a USI? So USI actually it's a path, it's a virtual path where you can gain information about your MS spectra. So let's begin the hands-on on reverse metabolomics. So as we are aware that we don't have internet access on each and every laptop, so hopefully like you will be able to follow later on. But first thing what we need to do is like we need to first get our MS spectra, and here like you could go to the library in GNPS. I don't know how many of you have used GNPS before, but if you click in the link, hopefully this will work here as well.
And maybe if there's no internet in some of the rows, just move to people who do have internet. And then, yeah, it's anyway better if you talk a little bit while doing the work. There will also be some tasks in the end that you can do together. And yeah, it will be always easier if it's together.
Yeah, and good opportunity to network as well. So basically click the link and you will go to this library explorer in GNPS 2. And first thing what we need to do is like we need to search for our compound, that is phenylalanine CA. So you could just go here and then type and then hit enter. So hopefully it works. So what you will get is like you will get I don't know, like something weird happened. So yeah, so when you search for this phenylalanine, so you will get the I guess everybody is following. So here you will see this kind of window.
If anybody does not follow, you can also just raise your hand. We are enough people, we can just like run around and help you on your computer.
So did everybody get here? Okay, good. So if anybody need help, you can just raise your hand, and then I guess one of you could go and help there.
Basically for our system, the internet is very slow a little bit, so it is taking time. So even if I've been connected with cable, so it is fast, so you have to slow down.
Okay, let's take it in a little bit.
How many people are waiting until it's loaded? Who got already the phenylalanine cholic acid? Ah, okay.
Okay, so
most people got it.
So you basically need to go to this compound, and then you need to filter the phenyl. Yeah, so you just type.
Use the slides.
Yeah. Yeah. So you could just go to the slides from that URL as well.
Yeah, just follow the slides
Yeah.
here, and then we want everyone to use the same USI, and the reason is because we are a big number, right? And we don't want GNPS to crash, and especially with the with all the internet connection.
Yeah.
So here, what you type in, you're gonna have different adducts, right? So we want the M+H of the second page.
Yes.
So please just go to the second page of all the results, and then the last one should be the one that you use. It has been deposited by Emily Gentry, and it's gonna be the USI that is indicated also here. On the slides. So you want to use the one that is that has the 2001 at the end, and then copy paste that USI. And then when you get there, then we're going to use it.
Yes.
But just please use the same USI to
Yes.
make sure everything is more fluid.
Yeah, I was just waiting. So I guess everybody got it there. So can you raise your hand, like who was able to get to second page and get the USI? Okay, quite a few.
Anybody still trying?
I guess internet is slow. I guess I will just move on. Yeah. So as Vincent was pointing out, you need to go to page 2 and click the last one. And then click on the circle. And if you scroll down, you will see the USI for this M+H phenylalanine conjugated cholic acid. And in order, when you click that that USI, it will take you to the Metabolomics spectrum identifier resolvable. So it will look like this. So this is where you can see your MS spectra. And basically, like, your MS spectra will be like displayed like what you see here. And next step is that we are going to do the fast search. So for the fast search, what you need to do is, like, you need to copy this USI and then go to the link fastgnps2.org fast search. So if you click to that link, it will take you this to the web page of the fast, and then you just paste your USI here where it says spectrum USI.
If your compound of interest doesn't have any USI, there's this indication here, right, that click to enter the peak manually. The reason why we prefer USI is everything is faster, right, and the precursor information is already embedded there, the charge state and everything, right? But if you don't have the USI, you can also generate one by just depositing your data into Massive, and it will automatically create one. We have a very well-defined documentation on GNPS on how you can upload your data to create the USI. We're not showcasing that today because it's already a lot of information to process, but it's possible to do both ways. So either you enter this manually, right, as a 2-column where you have the MZ value and the intensity, or you use the USI here. And for the second step, you have to choose the library name. There's multiple library there, so there was recently an update. So there's also one that it's called 2025 in the name, so just use that one. Which is the newest version of it.
Yeah. And other parameters, today we are keeping it default as it is. And then you just click on search here. So that will lead you to, like, if you, when you click search, you can scroll down and you will see the similar screen like as I'm showing at the moment. So basically, like, this is what you would need to export, but beforehand, like, what you need to know is, like, there are different informations. Whatever you are exporting here, like, you need to check that the delta mass, usually it should be 0, and this is the USI that is matched when you do the fast search. So basically it's matched to all the data sets that all the MS spectra in the given USI, and you will have a cosine similarity score. So if when you see cosine similarity score of 1, that means it's a perfect match, and then depending upon how many matching peaks there are, the cosine score can change accordingly. And you also get the information about the data sets where this MSMS spectra is, and that would be kind of crucial for you to know, like in upcoming step, like to match with the body part and other things. So basically what you need to do now is, like, you need to click export, and then it should be downloaded automatically. So depending upon where your download folder is, the export should be in CSV as a CSV file, right? So basically, like, it will be as a CSV file, and then you need to rename that to phenylalanine conjugated CA, like PHECA. Is there something more that we need to do? Yeah. So are we able to, like, how many of you are able to get here? Oh, quite many. Good.
Anyone lost any of the steps and needs help? Nobody. Great.
So basically, like, now what we want to do is, like, so for this workshop we are basically focusing on 3 molecules. So we did it for phenylalanine cholic acid. So basically we want to repeat this for phenylalanine C4 and then histidine C4. So you could basically repeat all these steps which I explained you earlier using this link. There are USI for each molecule. So you could repeat the fast search, export for each molecule, and rename it as written here in the slides. And I guess now, Vincent, you will take over. Yeah.
We'll just wait 5 minutes so you can search all the other 3 molecules and give you enough time.
I have one question. When you're searching fast in GNPS, so there is a lot of A plus H, M plus H. Which one you have to select? How you
We just have multiple spectra deposited for the same molecule, right? So the idea is to get the most, the more representative ones, right? And the one that have a lot of usually peaks, right? And good intensity, because it will also, and like, you're gonna have different matches, right? And you can also have a lot of false positive. If you have only 2 peaks in the spectrum, then for sure you're gonna get your false discovery will be a little bit higher, right? But if you have a good intensity, a lot of peaks, and then you have, like, for example, here you have 14 different match peak with a cosine of 1, then that would be good, right? So usually you should do a manual inspection of your MSMS spectrum before choosing it.
Yeah, it was a great question, actually. We just repeat it because I think it's valuable to keep in mind. So the question was basically how you select in the library what spectrum you should take, because sometimes you have, for the same molecule and same adduct, more than 1 spectrum, pretty often actually. And what's the right one? That actually depends very often on the context, right? Like, some of the spectra are generated by TOF, some are generated by Orbitrap, different collision energies, different noise levels. It's all very different, right? So what you generally want to do when you really care about the distribution of this particular molecule is definitely what Vincent just said, like, look at the spectral quality, because it's public data, right? Not all are equally useful. And then also try multiple, right? Because if you match against public data, some of them are also TOF, some are Orbitrap, different collision energies, right? So you wanna try different things and then investigate all the matches you get in the context of what spectrum you used. Okay? Hope that makes sense.
You also have in the article that we published, we also have, I think in box 2 or 3, it's like how you should interpret and how you should choose an appropriate MSMS spectrum. So it's also all in the article how you should process to assess it. Yeah. Did you guys retrieve all the 3 different tables for the 3 different molecules? Not yet? Okay, just wait.
Vincent, you're using neutral molecules here. You mentioned at the beginning about the diamino compounds, the polyamines. In their spectra, you get a very small 337, almost insignificant, because the charge is on the side chain and
Correct.
not on the
Yes.
not on the molecule. Does this work?
Yeah, I
Would
think
you-
it still works, right? Because Yasin can explain a little bit more, but the way the fast algorithm will work, it will select like the most intense peaks into a specific bins, and then in that case, you're gonna get, let's say 6 or 7, 8 match peak compared to like, let's say 14. But the way this algorithm works, actually should still works.
Yeah, because you're using cholic acid, so cholic acid will always give that 337.253.
Yes. Yeah.
But if you're looking in your samples and you've got a low intensity of that spectrum, you really won't capture that iron. You'll see the dehydration products, and that's about all.
Yeah, we also have in place different strategy now that you can also make sure that these are there in the MSMS spectra, even though the intensity is below, let's say 5%. But we also have these pulse filtering that will be releasing soon, actually in the available to everyone, that you can also have a second step of filtering, make sure those peaks are there to make sure these are truly bile acid.
Yeah.
And that will be there.
But it's important to keep in mind that at this point, like everything that you see here now, that's just based on spectral matching, cosine matching, right? So it does not check if specific diagnostic fragments are there or not. They might be or not. As long as they don't affect the cosine too much, it won't be considered for this match, and you will get a hit whether this diagnostic fragment is there or not. There are other strategies where you can consider that, but that's not what we are discussing here now in this context.
Yeah, I've been playing with MS Style, and it throws out a lot of data that claims to be bile acids, because I've got a bile acid conjugate library, and a lot of it, as I've got a post from this list here at the meeting, and most of it's rubbish.
Yeah, I didn't know that, but good to know.
Anybody needs help?
Just
The
internet
internet is
problem.
slow, yeah. Okay, who got it already? Anybody's internet fast enough? Okay. A few people, okay. Who is waiting just for like, okay, it's the majority, let's wait.
Let's wait. And then I think for the code, the script part, we were just gonna go a little bit faster because you need to download R for those of you that don't have it, and then download redo, so it would take some time. So we're just gonna go through, so you can just follow along, right? And then repeat it, the same thing yourself when you're gonna get good internet access. And we're gonna be here right throughout the whole conference, so you just want to grab one of us and then we can just go through with you.
And maybe very quickly, who has R on their computer? Okay.
Oh, good.
Okay, for those who don't, maybe also here, like, move next to somebody who does have it. Because I think downloading R will not work with the internet here.
I agree. Just waiting 5 more minutes, 2 more minutes maybe.
In the meantime, while we are waiting.
While we're waiting, can you guys comment on the libraries, GMPS, data, index, and all of those? What differences do those make in terms of querying with MAST?
What difference the library makes, or?
Can you guide us in terms of what library we should be searching against? Because it's a pull-down menu.
Oh, yeah. Yes, maybe while we are waiting, can we go to the?
200 live?
Not to the slide, but to the. I think we have it. Yeah, you mean here, right? Yeah. Yes. So what this represents is basically different indexes of the raw data, because the way MAST works is that we have downloaded all this public raw data. We then indexed every individual MSMS that we have in there. That's why we know how many MSMS are out there in the public space. So we really check for each individual one which peaks are in there. And depending on what data we downloaded, there are different things you can search, right? So if we index only a certain set of this data, then you will only be able to get hits against that certain set. And what set we selected is what's represented here by this library index. So the most up-to-date one typically is this one here. The nightly one, that one should update, like it used to update every night, so that's what's called nightly. At this point, the intervals are a little bit bigger, it changes from time to time, but that's generally the most up-to-date one.
So for a first-time user, let's say they're interested in lipids or amino acids or alkaloids, how do they know which one to select?
I see. Yeah, it's a bad naming. So the library here doesn't mean it's a molecule library. Library is just like what raw data have been indexed. So what raw data are you searching against? When you put up here a USI to an amino acid or a lipid, you will just search it in this case here against some subset of all GMPS data. Or if you put this one here, then you will do pan repository analysis, so you will search that lipid against all the repositories, right? And if you then put an amino acid up here, you will search the amino acid against all this data. So it's called library, but it doesn't actually mean a spectral library. It's more of a data science, computer scientist term in this context. Answers the question? Yeah, so you're not, okay, great. How many are still waiting? Now we are down to 3 people. Okay.
Okay, maybe we'll start proceeding then. So the next step is to get the code from the GitHub. So you have the link here. So you can just go to the link here, and then you're gonna see the reverse_metabolomics.r. So you have to select this one and not the one that it's from Python, because for sure you'll have problem throughout this workshop. So please, the one that it's finished by .r here, and then you can download it here by pressing on this button, and then you can open it into your RStudio environment. So I think this part, it will be probably a little bit more challenging. So please don't hesitate to raise your hands. We have multiple people that can help you here. So the code, we try to make it as easier as possible, but it's still coding, or it's still a script, right? So the idea is just to define 2 different directories, and then from there you can launch the code automatically, and then it should run correctly and without any problem. But defining those directories sometimes is a little bit challenging for different people, depending on your skill level. So please don't hesitate to just raise your hand at this point. So did you all get the link works, and then you downloaded the script already? Yes, for most of you? Good. So for here, you just have to choose the directory, and you choose the directory. So all the files that you downloaded, right? So we went too fast. You query, let's say, phenylalanine cholic acid at those 3 same molecules. You generated the 3 CSV files, and they should be into your download folder. What you can do with it is just to drag and drop those into your desktop, right? And then you can just have a folder. So just create a folder on your desktop, and then call it, I don't know, workshop. And then open that folder, and then create another folder in it. I just like to keep things separate, right? So create a folder, create a subfolder that you can just call fast search or whatever you want, and then you put the tree table that you fast search, and then you drag and drop into that folder. Make sense? So it's just 2 folders, and then you put the fast search into the second one, the subfolder. And then the first thing will be, if you don't have those packages, please uncomment those lines. So the install packages, data table, tidyverse, and pheatmap, just uncomment them and then run them individually. So you're gonna install those 3 packages, and then with the library, you're gonna load them. So please, if you have you're stuck at any point here, or you don't fully understand, just raise your hand and we're gonna go through you. We have different people that can do that. So hopefully you make it here. If you need more time, let me know. Otherwise, I'll just proceed. But here you have the folder path. So the folder is just the workshop folder that you created on your desktop, right? So it's the first folder that you created. And what you want here, so what we usually do here is we gonna automatically download the new redo metadata. So redo, it's uploaded like constantly, right? So we always want to use the last version of it. So the first time you're gonna use it, in theory, this part of the code here will download into your directory automatically. So you don't have to go to pen redo, you don't have to click on download, you don't have to drag and drop nothing. It will do that automatically and the script will work. But the problem with the internet today, I think most of you will not be able to download that fast enough. So if you just go back to the GitHub account, we did already put a zip file for the redo metadata. So here on the main page, it's not highlighted here, but you should see the redo metadata or all information with a specific date on it, and this will be the redo metadata. So just download it. It's going to be a zip file, unzip it, and drag and drop to your first folder on the desktop. Otherwise, I think we'll have a problem of just downloading this file. So I know it's a lot of like manipulation, right? But when you're going to get used to it, all this workflow of going to GNPS library, checking your molecule, doing the fast search, and running the script, it's a matter of like 2 minutes, basically. So when you get used to it, it's like super fast. I just know it's a lot of information to process, but usually we do that constantly, right? And it takes less than 2 minutes to do this whole workflow. So it's normal and now it's taking time.
Which file am I looking for?
For the redo metadata?
Yeah.
It should be like on the main page, on the GitHub, and then it should be, maybe I'll just access it and show you live actually. Oh, sorry, it will be here. So if you go to, so this is the main page, right? And then you have the fast mass search. And then here you have this file here called all sample redo November 8, 2024. And then this is the one that you want to click and to download. Good, nice. So you just put this one, you unzip it, and then you just drag and drop that one into your first folder. And then we just want to keep the CSV file into the subfolder, right? And this redo, you can just drag and drop into the main folder. So that'll just keep things separate. So the path that I'm mentioning are actually here. So the first folder path is just the main folder. And then yeah, you can just put your files there, and then the file list here, the first part of the code would just retrieve automatically the CSV file that are in that specific folder. So that might be where it will be like some issue. So please, if you don't see 3 files into your environment on RStudio, please just raise your hand, and then we'll just come to you and then fix the path. Can you just go? There's a hand, please. Thank you. So how many of you did get the 3 file loaded automatically? Not a lot yet.
I guess you don't have it on.
No, I don't have it, but you can get it from the GitHub also you want, but it's also not working. Okay, you can. Yeah, sorry about that. Yeah. I do have, but I just don't have the. You can do it. Yeah, yeah. Yeah, she can. Oh, just copy it. Got it. Yeah, yeah, yeah. It's a tree file, the directory to the fast file. So the first directory is just pointing towards the subfolder. So how many of you did get it now? The 3 different files, did you all get it? Just a few of them still? Yeah, then we can wait 2 more minutes maybe. Because once the first part is set and you have those 3 files and the redo it's loaded, then you know we're going to go step by step, but at the end you can just run this automatically and it will generate all the heat maps. At the end, right, if the 3 file are loaded, if you see the 3 file CSV file in the environment and then the redo is loaded, then they can do command A and then run this automatically and everything will be done, right? So, and then I can go to the step, but it's just for you to know, right? It's like as soon as they define the folder path, which is the subfolder inside the folder with the CSV file, that's it. Subfolder. Then the subfolder. You have a folder, right, that you create, and it's all on your desktop. You have a folder, right? Let's say you call it workshop. You will define the subfolder. Yeah, yeah. So you create a subfolder called the path, right? Yeah, yeah. I mean, at the end is because like sometimes the naming are changing, right? CSV, TSV, and then you want to change like to CSV or whatever. So I want to keep them separate. So then even if you change the name, because the TSV is the redo, right? But if you come to a CSV, then the code will not work, right? So if you keep them separate, it will always work. So that's why it's a little bit more challenging. But it was just like everything changed all the time, right? So we never know. Because I thought of putting everything right and then say the CSV file would be loaded, but yeah, I know. Yeah, yeah. So how many of you did get the 3 files now? Is this more than 10? Please raise your hand, because otherwise, like, there's no way I can know if I can proceed or not. Okay, just a few of you still. Maybe it's internet problem. Who has still an internet problem? You? Okay. I mean, I can go slowly throughout the different steps, right? But as soon as you have the detection of the 3 files, right, that is your subfolder inside the folder, and then the redo, you have drag and drop into the first main folder, then you can just hit Command A and run this whole script automatically, and it should generate 2 different heat maps, or 3 different heat maps actually, where we have the body distribution of these 3 molecules. I'm just going to go through the code, so then instead of just pressing button, you know what we're actually doing. But this is just a basic script where you can customize probably everything you want, right? But it will just at least give you different body part distribution of these 3 different molecules, and if it's associated with specific disease. So the first step, right, is just to load. So you have the folder path here. So it's the directory to the fast file that are in the subfolder of the main folder. And then you create an object, which is the redo metadata, and this code in theory will download it automatically. So when you have high-speed internet, it's not going to be a problem, and it will create and put directly this file into the right proper location, so the code works perfectly. And then you gather all the file, you read the file, and then you combine all of them. So that's why we renamed the file. So when you do a fast search, you say, okay, I have this USI, I search it, and then you download, but it will download like data2.csv. You rename that file phenylalanine acid, so you don't interfere with all the different molecules that you're going to be querying. And the code, what it will do, it will take the file name automatically, let's say phenylcholic acid, right? And then it will attach this and create a new column. So this is the column that you have here. So we'll create a new column called compound, and it will list the file name that you choose. So if you call it histidine C3, it will take that name and fill that column with it, so we know which USI it's matching to which compound. And this is important when you generate the heat map to know which compound is associated with which biofluid or body parts, right? And from here, we have to, once you did the fast search, so the fast search, it takes your USI of your molecule of interest, your unknown feature or known molecule. And then it will match and it will collect all the different USI from the public domain that is identical or similar based on the cosine and matching peak, right? And then what we need to do after that is to integrate the metadata. So when you deposit the metadata, it's at the file name level, and when you did the fast search, it's at the scan level, right? So because it matches exactly the same MSMS spectrum, so we need just to harmonize those two tables so we can actually join them together by a common identifier. So what we're doing here in the second part is just to modify a specific column into both tables that we call USI to make sure they're matching. So some of them will just collect the massive ID, the massive number, and the file name. And we remove all the rest that we don't need. But this is doing that automatically by just launching these lines, right? And then at the end, what you're gonna get here is a combined table where you're gonna merge all of them. But just before we do that, we just harmonize some of the naming again. So the first part here, where you have standardized here, if we combine all the skin location to skin. So we have skin of head, skin of trunk. So for us, we just want to combine all type skin together to skin. If you're, of course, if you're interested into a specific skin location from the redo metadata, then you want to remove that line. So you just comment it, right? Because otherwise it will comment everything. It will aggregate everything to skin. Same for blood. So if you have plasma and serum, we combine them to blood. But if you're interested in a specific molecule in the plasma or the serum, you want to, again, comment that line. And then we just do some formatting here. And then we separate two type of organism, right? So on one side, we want only human data. And on the other side, we only want mice data. So we just filter, and then we keep only the USI that will be matching to human, and only the USI that will be matching to rodents. And for all the rodents, we just, again, aggregate all the different taxonomic ID that we have in Redo into only a single list of rodents. This is how we're doing it. Of course, if you're interested only in mice and not with rats, you don't want to do this. But for the first overview of the data to see if those molecules are mostly associated with human or rodents, then I just recommend to do it as it is. And then when you get comfortable, you can just modify whatever you want. And then you're going to have a data frame look like this, and where you only have information on human, for example. So here on the complete right of the table, you're going to have the NCBI taxonomy, and only human information will be embedded there. And it's the same for mice. And then what we want to have is the count per organs. So we just want to know how many times this spectra is found into specific organs. And you do the same for human and the same for mice. And then you're going to have a table that will be generated that looks like a summary table, where you have, okay, I have feces, I have 619 matches, and it combines all those different molecules. So I have histidine C4, phenylene C4, and phenylene cholic acid. And then here is just to reformat, right? It's just modifying tables to make it compatible with the PEAT map package for data visualization. So it's just formatting, and just, again, more formatting, classifying everything into alphabetical order. We place the body parts, so the data frame only need to contain its numerical value, but here you have the first column will be Uberon body parts, right? So then we just place that as row name, and then all your data frame will be composed of numerical values, and then we log scale, and then you should have these first two heat maps that will be generated, one for human and one for rodents. So here you can already see that some of these molecules have different distribution pattern, and you get a lot more matches on rodents, right? Of course, because we have access to more body part of biofluid than in human, and we have much more data on rodents than on human for different specific body parts, right? So for this, that makes sense. So if you reach that, that'll be super cool. So if you, I will let you 5 minutes, and just to launch that. But if you already get them, that'll be great. Otherwise, we can just come to you and help you fix it again. Yeah, I mean, I think you will have some time, so maybe at the end. So how many of you have generated the heat map so far? Oh, great. Okay, quite a few of them. Okay, good. So that was the first part, right? Just to generate the heat maps, and then it's body part distribution. It's kind of the raw counts, and then it would just log it, right, to just make a visualization better. So there's no normalization that is done here, although you're welcome to just add it, right? But we just want to get an overview of how these molecules are distributed across different body parts of rodents and humans. And then the second step that we were interested in is to find if those molecules are associated with specific disease. This is what we care for us when we discover those type of new molecule in different biological contexts, or if we have different modification of a drug, for example, right? Maybe this drug was supposed to target a specific location, but then it get modified by microbes, and then it reach another location, so you have off targets, right? So these are all the type of analysis that you can do. So the first step again is to separate again human and rodents data. So we do exactly the same with, and this we apply to the redo metadata, and then we do some filtering. So here we're gonna focus on the DOD common name, which is actually referred to the different specific disease, but the codes already can provide you with life stages, health status, or biological sex. So if you're interested into molecules that are they found only in men versus women, then you can also apply this. It's already kind of integrated into that script here. So again, when you do the same, then you just summarize, you manipulate the table, and then you again link redo metadata here, and then you should have this first data frame that is generated, where you have different disease that you have on the left, and then the number of counts that it's there. But here we want to normalize. And the reason why we want to normalize is that in PenRedo, we have much more data in IBD, for example, inflammatory bowel disease, compared to HIV, for example, or patient living with HIV. So what we want to do here is just a normalization step where you have the number of total counts for those specific disease, and we just want to normalize it. And then at the end, we're just gonna report that as a percentage. So when you run this, you just normalize, and then you calculate the sum, and then you just divide, and then at the end, you get a percentage of spectral matches associated with each of these specific disease. So here we already show in the past that phenylalanine acid was already associated with IBD, but here you get also the different subgroup, right? Ulcerative colitis and Crohn's disease, which kind of had confidence that this molecule seems to be found mostly associated with those diseases. The limitation, the challenge with reverse metabolomics is this is not causation, right? It doesn't mean that because this molecule is found into that specific cohort that it's necessarily meant that it's causing or it's involved in the disease, right? So here it helps you to formulate an hypothesis that maybe these molecules will actually be involved, or this molecule might be important as a new biomarker, or different, you can formulate multiple hypotheses, right? So what you need to do once you get that is kind of to get to the validation part, where we need to go back to the raw data, and then you start to do the stats on it. But at least this gives you more information that you previously had on these specific molecules. And this is what you get from here. So everyone was able to generate the last heat map here. So how many of you did reach that part? Okay, maybe I'll wait 2 minutes, or yeah, maybe I'll wait just 2 minutes.
I'm getting some error with one of the commands of the filter, that the delta mass is not found.
Can you go and check that?
I got the same error, and I don't know if the delta mass is supposed to be an object in R or...
Here, the delta mass is actually just the column, right? It's just, you mean at the first part where you filter?
Yeah.
Here. This should be just the column actually where you specify they should be within that range. Maybe it's because it's not a numerical value in your table. Yeah. Oh, do you have the tables loaded? Yeah. Do you have the table loaded?
Yeah, and the delta mass is not there, so I think it's one step before that.
But it should be part of the tables when you load them actually. So maybe you can check. Where I normalize? Sorry, can you repeat?
When the data is getting normalized,
Oh, it's here.
anything fully gets a sum of work,
Yes, it's here. So,
and then as part of the change.
should I go back or?
Yeah, the last normalization we just did a moment ago, which is normalized by sum, right?
Yes.
But I didn't
Here.
You didn't get the sum of work, sorry. The sum of the,
So this first table here,
yeah?
here, right? So here you just get all the different counts that are associated with each specific disease.
Yeah.
And then here we just normalize all of them, divide by this total number of count that we have.
Okay.
And then after that, you have to sum the different column to bring it to a percentage of spectral matches, right? So then the next part here, you just divide by the number of files that we have for each specific disease.
Okay.
And then from here, you just calculate the sum, right? So you can actually convert it to a percentage for reporting here.
Okay. Yeah.
Make sense?
Yeah.
Okay. Yeah. Oh, wait, I don't hear. Can you just go here, please? Thank you, thank you. Or maybe we can circulate it.
Great. Okay, I didn't know if that works. General question, similar to MicroMast, have you adopted a re-ranking or boosting process that, because you try to predict, let's say, producing organism, and if we see that the one feature is expressed in gut microbiome and the other is expressed in cyanobacteria, and then in microbiome, so it's like 0.9 cyanobacteria, 0.6 gut microbiome. Would you like boost to the 0.6 to go like to 0.8? Because probably if they're collected from the same sample, the one cannot be from cyanobacteria, the other from gut microbiome. So have you adapted, or would you like to adapt like a re-ranking or boosting of the possibilities to make more sense of the overall result?
So if I can repeat it to better understand it, so if you have a feature, right, that it's found in be produced in your gut microbiome, for example, and you have other ones that are found in cyanobacteria.
And you know that both of them, they're from the same sample. Like
Yeah.
if you have like 4 samples, that
Yes.
4 features that they come from the same sample,
Yes.
and the 3 of them, they're from gut microbiome, and the one has like cosine 0.9 that it's from cyanobacteria, and 0.6 that it's from gut microbiome. Would you boost like 0.6 to meet the others at like 0.8? So would you like re-rank the?
Yeah, you can play with that. I think it depends. I mean, you get sometimes those matches, right? And then sometimes it's better to. So it depends how your molecule will fragment, right? Because sometimes with the more peak you have, then your fall discovery rate will be much lower, right? But sometimes you only deal with 4 peaks, right? And then you can have a higher fall discovery rate. So in that case, you can say you can boost your cosine score definitely. You can put 0.8 and over 2 or 3 or 4 match peak, right? That you can also add that to boost your confidence. But again, you have to inspect the MSMS spectrum, right? You have to inspect it, and does it make sense biologically that these organisms will produce it? If it doesn't make sense, then it's maybe just a false annotation. It's also possible, or sometimes those peaks will have a higher delta mass than 0.05. Or it will be 0.05, but I would not necessarily trust it if it's a high-res mass spec. So I think you just have to always question and question your data, right? But the manual inspection of the matches is actually very, very important, depending on the class of molecule that you're doing.
Okay. So it's up to the user.
Yeah, it's up to your user. Yeah, everything is modifiable, right? You can apply 0.9 cosine score if you want, and then have over 10 match peak. But then you need to know how your molecule is fragmented, right? For bile acid, it will make sense. But for NaCl lipid, if you have histamine C2, for example, then maybe having 10 match peak is unreasonable, right? And you won't get matches. But if you need to know, you need a little bit, a background on how you do things and how to be cautious with it.
Okay, thank you.
Is it working? Okay. But what is the common problem?
Like sometimes the spaces are not the columnates.
Oh, okay. Guys, there's another question over there. If you want to go. But how many of you so far got to the last heat map? Oh, that's good. Okay, okay. You still have a question over there on the left? Do you have a question? You good or? Okay, we will just go ahead now. So if you don't reach this, the last heat map, you can still try it. But I'm just gonna go ahead and just finish this workshop by showing you how to use micromass, which is much faster. And just, it's a whole interface, right? So it's a little bit easier. But again, you have those 3 molecules, right? So now you get body part distribution in human and in rodent. So now you know, okay, these molecules are reaching different location, but are these molecules found in specific monoculture of bacteria, or is it plant-derived, or is it food-derived, or eventually we're gonna have microbiome mass here, which also is gonna be much more informative, but for now we're just gonna showcase you microbiome mass. So we published that paper a year ago now, and it's the link here that you can get access. So basically what it's embedded into that infrastructure is 60,000 LCMS run of monoculture. So it's only monoculture, and if you use that interface, then you can say, okay, it's found in a monoculture. You just need to be careful, because here it doesn't mean that, I mean, when you get a match, it's like, okay, it's found into that monoculture, but if you don't get a match, it doesn't mean that the microbe is not producing it, right? It's just, maybe the right microbe who's produced this specific molecule is not into that specific database. So you just need to be a little bit careful on how you interpret this data. So when you go and you access that link, you're gonna get to that interface here. And again, everything works with the USI, right? Because it's just faster and you don't have to provide the precursor mass and the charge again. So you just copy paste the USI and then you put it right here in the spectrum USI, and then you keep all the other parameters by default. Here you can also refine those parameters eventually, right? So if you want to go lower into the fragment tolerance, or if you want to go higher in the cosine score, you can also modify, but just for now, for this presentation, just keep everything as it is, and then you have also the number of match peaks that you want. So if you're creating bile acid molecules, we know they have a lot of fragments, right? So then I can go to 6, 8, even 10 peaks to have a good match. So that will increase that number. But if you're working with the small, very small molecules that the MSMS spectrum doesn't have a lot of peaks, then it doesn't make sense to go to 5 to 10, right? So you have to keep it low. And then you have to do the manual inspection to see if it's a good match or not. So I would just recommend to know how your molecule is fragmenting to make sure that the results make sense, right? And then when you're going to query, let's say this phenylalanine cholic acid, you're going to get this kind of tree that's going to be displayed exactly like this. And then it can tell you, okay, this molecule is found into some bifidobacteria, Enterococcus, lactobacillus, and we know that those specific bacteria, they do have a BSH, right? So the BSH is biosalt hydrolase. We know this gene is able to conjugate phenylalanine to cholic acid. So then it makes sense in the biological perspective, right? So if you're querying a drug molecule, a drug metabolite, and then you found it here, maybe that doesn't make sense, right? Because why this drug will be suddenly showing up into this infrastructure. So just be careful on how you interpret spectral matching, and does it make sense or not. And we also have now, besides micromass, right, we also created this new tool now that is called tissue mass. And here it will get you biofluid distribution. Again, it's a little bit like reverse metabolomics, but visualized into a different way. So here in this paper, we just confirmed 2 new molecules, which are 2 different carnitines derivative. And then we wanted to map their distribution rapidly and see if they're associated with specific disease. So it's the same thing again. So you have one specific USI, you just put it in tissue mass. It's the same, I don't provide the link here, but I can add it. It's just tissue mass again. And then it can show you, okay, it's found in specific disease, it's found in urine samples of human, it's associated with diabetes. So this is just a similar tool that you get to use here. The code that we did together today, right? So you were using one molecule at a time. So you use one molecule, you collected the USI, you retrieve all the similar identical spectrum from the fast search, and then you download it and then create this, right? And then you repeat that for the second molecule and the third molecule. But let's say you're interested into a full class of molecule. So if you want to query thousands of molecules or hundreds of molecules, you cannot do that one by one, right? It's not gonna make, it's not gonna work, or it's gonna make, like, it's gonna take you days. So we just have this script that we also uploaded on GitHub that it's right now available to you. And this one will allow you to create this same heat map or the same workflow that you have, but with tens, hundreds, and thousands of molecules. The only thing that you need is actually just a Google sheet where you have USI and then you have the compound name, and that's it. And then you can go to a specific GNPS workflow that is called the batch fast workflow. It's also available on GNPS too. You can add it to the documentation, but know that the script exists and there's information embedded into the script here that tells you how to proceed with it. But know that it's possible to achieve what you just did today with tens to hundreds to thousands of molecules. We make it super efficient. And yeah, with that, we'd like to thank Peter Dorenstein, of course, my mentor, Ming Wang, that is creating and maintaining all the GNPS2 infrastructure. So if everything is possible right now, it's because Ming is taking care of all the infrastructure. And if you want to submit your data, if you want to contribute to any of these tools that I just mentioned to you, especially microbiomass that we're actually creating right now, and I think in the next 2 or 3 weeks we will release it, even though we don't have any preprint yet, it will be available to you to use. So please just find us. And we have different talks and different posters where we put in practice reverse metabolomics on how we did that for thousands of molecules and what is the type of discovery that you can make with it. So I have one on Wednesday on how we discover a new molecule and how we were able to find that it was associated with inflammatory bowel disease. We have Santosh presentation here on different microbial metabolites and a poster, and then Yasin will talk about in-source fragment on Monday. We provided a lot of the links here, and all the different masks are available here. So tissue mass, micromass, plant mass. We have a personal care product mass that is actually right now available to you, where Yasin actually collected a lot of the personal care product to see if those molecules can be found into different product, right? And then we have food mass. So please don't hesitate to play around. Like I said, I think today was, it's still heavy, right, with the code, but when you get used to it, it's a matter of minutes, right, 2-3 minutes, and then you get the whole process and you get that whole picture, and you can reveal much more information about all those different molecules that you've never done before, right? So if you're also interested, we have some different molecule here, different questions that you can address. There's a lot of times remaining, maybe 20 to 25 minutes. So if you want to play with it, we have some different questions here, and we can also help you to fix any of the remaining problem. What I want to mention here is the first point. Maybe I'm just going to talk about this. Yep. Oh, yeah. We have another thing to show you before you leave that maybe will make your life easier that we were just able to accomplish with project in Train overnight. But before I would just want you to focus on this specific molecule, so histamine C5. It's a recent discovery that we made in the lab, and then it's part of the Cell paper that we just published like I think 2 weeks ago. And this, when you query this specific molecule in micromass, you get no hit. So there's no result at all. If you search again tissue mass, you're gonna get some hits. But then if you don't get matches, it doesn't mean that this particular molecule is not made by microbes, right? It may be just because the substrate is not available. So histidine, you have to decarboxylate histidine to histamine first, right? And then after that, you have to make the second step where you conjugate histamine to C5. So if you bypass the first step, right, if you provide histamine in the media, then we reveal that a lot of the gut microbes can actually conjugate the histamine to C5. The only reason why you don't see it is because probably the same, to achieve that, right, there will be one microbe that will decarboxylate histidine to histamine, and then a second microbe that will click or conjugate the histamine to a C5. And then we detect this molecule only when we cultivate microbial into a community. So if you have 10, 20, 30 different microbes as a community, then we start to detecting those specific molecules. And this is what you will find into microbial mass, where we have all these synthetic communities. You can reveal different function that the microbes can do when they're together compared to when we are alone. So here we confirm that actually this one is microbial derived, even though it was not detected in micromass. So that's the only limitation, that the challenge that you have is just to make sure that you interpret your result correctly. It's not because it's not there that it doesn't mean the microbe cannot produce it, right? And then we have an app that Project will explain to you. So we created an app that makes this script a little bit easier, where you just drag and drop, but he's going to talk to you about it for maybe 10 minutes. Oh, yeah, it's here.
Yeah, and then do you have the files, like, just easily accessible? I think you might need to just stay here to upload.
They're on the GitHub, right? But
or do you have them on your computer?
I don't have, this is not my computer, right? So I don't have it.
Yeah.
The files I don't have.
Yeah. Let's see.
I just show the,
GitHub.
they have the files, right? So if you share the link with them.
Download. Yeah, I think maybe we can just download the raw.
You can also explain them while you're doing this.
Yeah.
All right, so I think a lot of you probably had issues while running the R code earlier. So we just recently made an app to hopefully make things easier. I'm just going to pull it up right now. And right now I ask that, like, ideally, like, nobody try to visit this link because we're hosting it on the free version of Streamlit. So if everyone tries to visit at once, it'll probably crash. So I'm just going to demo it like live right here. And then when you're all using it on your own, it should probably be fine. So I'm just going to take the fast files that, like, from GitHub that we were using earlier. Shift like to select multiple files.
Oh, it doesn't really
How do you do that?
Can you do it one of the data?
I think it would be good to have. Is there no way to select multiple? Because
If you just press it and then you just create a square one.
yeah, maybe just one by one. I think we'll have to do that. Okay. So it's the same 3 files that we were using earlier. And then now I'm going to just select at taxa options. We'll do the human again. And then now we'll just let it run. It takes a couple seconds because it's loading the full redo table into memory. And we can again visualize any of the metadata columns. We'll do body part again. And here you can just select which variables you want to include. So if you want to limit your analyses to a certain subset of variables, that's possible. And then you can just submit, and then you get the heat map like this. It's a little zoomed in, but this should hopefully make your life a lot easier. One thing you'll notice is that it's slightly different in that we don't collapse the blood into one category here. That's just something that is more doable on your own, but as is, we just keep the redo metadata variables as is without collapsing them on this app. So this is just more meant for initial exploratory analyses, and it should be a lot easier than running the R script. Full disclosure, I coded this just starting last night and just finished 10 minutes ago, so it's probably a little buggy, but we'll be pushing bug fixes for this within the next couple weeks. So if you run into any errors right now, I would just encourage you to just refresh the app and try again, but it should be functional and we'll be updating it in the next few days, few weeks. Yeah, thank you.
Okay, and we only have 5 minutes left, but if you like, all these things that you have here on this slide, you should be able to do now. So if you want, you can just pick out a few of the tasks. You won't get through everything now, and try to get these things done. And if you run into problems, you can just ask us. But yeah, you should be able to do all of these things based on what we have talked about now, and answer these questions by navigating these different links that you have on the slide before. You should be able to, answer these kind of questions. So either you can try to go through some of these tasks, or you can just ask us questions if you have broader questions or specific questions. And we have at least one.
So thank you so much. It was very interesting. How about the metadata? So one can just download all the metadata associated to every particular study, like observation per observation, and there is all the associated metadata and can be downloaded through this also?
Yes, you can download all the metadata that, through Redo. There's just a button, download all metadata, and then you have it all locally, and you can do whatever you like with them. Yeah, it's about, I think, 700,000 files at this point.
Yeah, okay, But, very useful. Thank you.
Sure.